Okay, so my name is Serghei Mangul. I'm from RNASeq lab. So I think I told this joke a couple of times before, so if you heard it, I apologize. So this is what the title of the talk is. So it's dumpster diving. And actually, there is a definition of dumpster diving, so which is the practice of looking through the commercial resident garbage in order to find some food, which is perfectly fine to eat, or some clothes, which is perfectly fine to wear. So I'm not here to convince you that that's the lifestyle to follow, but we can borrow some ideas from dumpster diving, meaning that we should be always careful about throwing the data. So if you think about RNA-seq, given in RNA molecules, you will shred the RNA to the fragment, and you will sequence the fragments. So given the genome available, you would try to map the reads onto the genome, where the fraction of your reads will map to exons, and another fraction will span the exon-exon junctions. Some reads will go to introns and intergenic region. So given that genomic, like the profile, you can find the gene expression and possibly find the differentially expressed genes, as well look at the RNA splicing structure, and if you're sophisticated bioinformatician, you can try to assemble the full-length transcript. So given all that, you will never consider the reads which were not mapped to genome, so they will go to the trash. So we don't think that's the right practice, so we designed what we call the read origin protocol, which takes care of the unmapped reads, trying to carefully examine every single read. And if you want to know the details, the paper is on the archive. So given the unmapped reads, we are able to find the bunch of classes. And first of all, we were just curious, can we explain every single read RNA-seq provides you, as well as does it provide a possibility to answer new biological questions which were impossible to answer with the mapped reads. And then just going kind of a little bit in advance, I will tell you it does. So for example, you can quantify the immune response or study the microbial communities purely from the unmapped reads. Okay, so we started the mapped reads, where we will classify those into the genomic regions, and as well as into the repeat regions. But again, so there is a 12, on average around 12% of the reads which we know nothing about. So as we thought about the protocol, the first step, we do the quality control, where we filter the reads which are low quality, low complexity, or the reads coming from the ribosomal RNA. So given that, we are able to filter out around 5% of the reads which are low quality and low complexity, as well as 2% of the reads which comes from the ribosomal repeats. Next, as we move on, we thought that all the high throughput mapping tools comes on the great speeds, but providing no guarantee that every single human reads will be identified. So what we do, we suggest to use the more slow but more sensitive alignment megablast to identify what we call lost human reads. The reads which are human in origin, but were missed by the original high throughput mapping. And then we are able to identify around 5% of the reads which are human reads, and then we are not identified by the high throughput sequencing. So if you're curious about how those reads are, we look at the edit distance of those reads comparable to the reference, and then we see quite a fraction of the reads which are within the threshold which Tophead uses, meaning that there is less than 2 edit distance of 2 comparable to the reference, as well as some reads comes with the greater number of mismatches or insertions or deletions. We also were curious to know if there is a specific location in the genome where those reads are coming from, and they are randomly distributed across the genome. So what we did, we took the mapped human reads and we classified those into the genomic categories, and that was poly-A selection data, where the majority of the reads are coming from the coding regions. And then if we also look at the lost human reads, and then we categorize those into the genomic categories. So you can see the profiles are quite similar with some exceptions. For example, the introns are largely, the fraction of the introns it's much larger than the fraction of the introns in the mapped human reads. So we did this as well for the ribodepletion protocol, where the majority of the things are expected to come from the introns, which holds for both mapped human reads and the lost human reads. So as we move on, we thought about the lost repeat reads, the reads which come from the repeats, but were not identified by the mapping protocols. And we used again the megablast to map to the database of the repeat sequences, and then we were able to identify the tiny fraction of the reads coming from the repeats. So the profiles, again, suggesting similar, the profiles, the profile of the lost repeats and mapped repeats are quite similar in terms of the, if you look at the ALU, in both cases the ALU are dominant class across the lost and mapped reads. The same was for the RIBO depletion protocols, where the majority of the things were coming from L1. As designing a NESTAB, we thought about the things which are not linear in terms of your genome, and things like coming from so-called non-collinear RNA, which is the circular RNA, the transplicing, or the fusions. So if you think about the transplicing or fusions, so you have a splicing which goes across the genes. So in this case, the exon 2 from one gene was spliced with exon 2 from another gene, forming a transpliced transcripts. If that occurs within the chromosome, we refer to it as transplicing. If that occurs across the chromosome, we refer to it as the fusions. As well as the circular RNAs, where the transcripts are circularized, and then we see the evidence of the splicing kind of in the tail to head fashion. So given that, we compare three tissues, the blood, nasal, and the lung, and then consistently and then the lung and the nasal, we use those tissues as a comparison, thinking that the transcriptomic profile of the nasal and the lungs are quite similar, providing us the tool to conclude that lung was prepared by the RIBO depletion protocol, and nasal was prepared by poly-A. So we conclude that the total RNA, as expected, provides you more reads coming from all three events. Unmapped reads also provide you the possibility to quantify the immune response, looking at the VJ recombinations from the immune locus, and as well as looking at the random insertions which are inserted between the recombinations. And then we're able to identify 0.02% of the reads coming from the recombinations. We look at a couple of tissues from the GTEx, where we observe the largest of the fraction of the reads coming from the spleen, followed the blood, lung, and other tissues. And then we compare the fraction of the reads coming from the B and T cells. So Ig, this is immunoglobulin, which stands for the B cells, and TCR, this is T cell receptor locus. And we see the majority of the things in the spleen coming from the B cells, meaning that the spleen secretes the B cells. As well as we look for the isotypes, where we look at the constant region of the heavy chain and looking at the relative abundances across the tissues, where for the mu, if you look for the mu isotype, it's highly expressed in the blood, and then it stands for the naive B cells, which is known that the blood has a lot of naive B cells. So given that, we look across all the GTEx tissues, looking for the number, the y-axis stands for the number of VGA recombination per tissues, and we see the highest number of VGA recombinations per sample in the spleen, followed by salivary gland, small intestine, which actually was designed by the GTEx to be the tissue of the lymphoid tissues, just because the part of the small intestines which was taken was in the close proximity to the lymphoid node. So we thought about how to measure the immune diversity. So given the VGA recombination observed in the data, what would be the best way to measure the combinatorial immune diversity? So we took the ideas from the microbiome. We are given two farms with the 4 species. They are present in the different relative proportions. Here it's uniform profile. Every species is like taking 25% of the data. As here there is one dominant tissue. So we use the alpha diversity measured by the Shannon entropy, and that, as you have the dominant species or dominant, in our case, VGA recombination, we will say that this is less diverse than this farm. So we use the beta diversity, basically trying to compare the compositional similarity between the farms. So we'll use these two measures to assess the combinatorial diversity of the immune system. We look at the since we have the case control status, we look to see how the asthma individuals are different from controls. And this is the nasal tissue. And on the kind of x-axis here, horizontally, you have J genes, and this is J genes. And the color, as more red it is, as in more sample, the particular VJ combination is present. And I guess even from the heat map, you can see that the controls being more diverse. There is more VJ recombinations being present in controls. So we take a formal way to assess it. So we took the alpha diversity per sample, and then we see a significant increase in the alpha diversity in the controls. As well as we look for the beta diversity, where we compare the controls to themselves, the asthma individuals to themselves, and the asthma to the controls. And what we see, so as lower the beta diversity is, as more similar individuals are, and we see the controls being more similar to each other than the asthmatic ones, and also the controls are more similar to each other than to asthmatic individuals. So this is a certain kind of profile being shared in the controls, which is quite different in the asthma individuals. The last step of the pipeline, this is the microbiome. So you can think of every sequencing as the metagenomics, just because as you sequence the tissue, you will also capture the microbiome harboring that tissue. We use different tools to assess the microbial reads, to extract the microbial reads coming from the viruses, bacteria, and eukaryotes. And then we often observe enough reads to infer the taxonomic profile, or sometimes even go to the functional profile. And then, given that, all the read origin protocol, we're able to explain 99% of the data, comparable to the 85 or 90% done by the mapping-based tools. So what is this 1%? Actually, we were surprised to see template jumping. What we did, we did look at those reads, and then sometimes we see half of the read coming from the human and half of the read coming from the worm. So given that the worm cannot integrate in your genome, that's clearly template jumping. As well, there might be unknown microbes. That's why with David, we designed a tool, EM which will do which will compare the microbial communities, not using the reference. Okay, so we run the ROP, and then based on the metaflation across the several GTEx tissues, and then so far what we observe are the taxa which is expected to be in the human microbiome, so we're happy with that. As well, we compare the alpha diversity across the tissues, and we don't see anything in the gland and nothing in the brain and the heart. So we're happy about this because we plan to use those as a negative controls to control the contamination. Currently, we do extend. We want to go beyond the bacteria, and this is the positive control which we see. So we took the LCL samples where EBV virus was used to transform the LCL. So we clearly observe the nice coverage of EBV. So given that, so kind of saying us that if the virus would be in your sample, the pipeline will be able to detect it. Okay, so given that, so we designed the ROP tool, which is able to profile the repeats, the non-collinear RNAs to quantify the immune response by looking at the VGJ recombinations and to study the microbial communities. The typical output of the pipeline is something like that, where for every step, we will tell you how many reads are falling in each categories. No installation is required. We distributed all the tools we are using, besides the tools we developed, with the protocol, acknowledging the tools we are using. And there is a detailed tutorial which you're welcome to look at, and then it has all the instructions to run. So we're exploring different technologies. So far, we look at RNASeq. We got some requests from Australia. Look for the chip to trying to run this protocol for the chip-seq. Single cells, this will be quite interesting for the protocol. There is future directions, so we're extending, and then with David, we're working to profile the bacteria, viruses, eukaryotes, making the standalone tool. Okay, so given that, I would like to acknowledge many people involved. First of all, as RSkin, Noah Zeitland, which we are leading the project. Serghei Mangul was providing inspiration and super helpful comments, so thank you so much.